Hi, this is Dr. Rhonda Fellheim. We're going to continue our discussion of the cardiovascular system, but we're going to be focusing now on blood vessels and circulation. Really, just, I keep using the word amazing and it seems like such an understatement. So I'm going to have to come up with a better word, obviously. First, what I wanted to do was take a look at the walls of the blood vessels. And so when we say blood vessels, we're obviously talking about arteries and veins. And what I wanted to let you know is that the walls consist of the same structures for both arteries and veins. However, the various layers are thicker in arteries than they are in veins. And so the walls of veins are going to be much thinner. However, the same layers are there. Within both structures then, what we have are three layers: tunica interna, media, and externa. And the tunica interna is going to consist of our endothelium. And endothelium is just that layer of cells that will be touched by the blood, or the wall that will be touching the blood. And then the tunica media is where we're going to find our smooth muscle, and we know that smooth muscle is going to control the size of this lumen. Okay, the lumen is the opening where the blood flows. And the way that it does that, smooth muscle will constrict, meaning, or contract, meaning get shorter, which will cause the lumen to get smaller. And then as the smooth muscle relaxes, the lumen will enlarge. All right, the outermost covering then, the tunica externa, what we're looking at is just a connective tissue covering. This is kind of fun. As we look at the heart and the vessels that come away from the heart, they start out very large and then they get smaller and smaller the further away from the heart and out into the tissue that we get. So, we have these, we're going to talk about different sized arteries and what their claim to fame is, but I want you to understand that these arteries are just super highways. They're just highways carrying blood out to the smallest of vessels called capillaries, and that it's at the capillaries where there's going to be an exchange that occurs with the cells. And so we know that the whole purpose of all of this is to get blood and nutrients out to the cells. And so the actual action between blood and cells is going to occur at the capillary level. So everything before the capillary level, everything after the capillary level is all just transporting blood. So, the analogy that I like to use is like, okay, so we're on I-75, which is going to be these larger arteries, I-75, boom, boom, here we go. And we're speeding along. We're just transporting blood is all we're doing. We're not having an exchange with the cell. So we're going up or down I-75, and so I'm going to get off on Williston Road, which, is well, let's get off on Newberry Road, okay, because it's going to be, 4 lanes, 2 going each way. And so even though we're smaller than I-75, we're still, a pretty good sized road here, moving the blood along. And where are we headed? Well, we're headed to our friend's house out here, but we don't get to interact with our friend until we pull up their driveway, okay? So we're gonna get there by heading out Newberry Road. Then we're gonna turn off on 337 South, and we're gonna drive down that road, which is a smaller road, until we get to the capillaries. And the capillary, well, what do we call that? We'll call that the driveway, because once we pull in the driveway and park the car, we get to interact with whoever it is we're going to visit with. Okay, so that's where the blood is going to have its exchange with the cells. So let's talk about some characteristics of the different sized arteries that we have. The largest of arteries are those closest to the heart. We refer to as the conducting arteries. And you can see all the squiggly dark lines that you see within the tunica media. And those squiggly black lines represent a fiber that you learned about in 2085. I'm going to help you out here. It's called elastic fibers. And we know that elastic fibers will stretch and then recoil. Okay? The fibers themselves don't contract, they just stretch and recoil. So that's going to come in handy, especially in an artery like the aorta. This very large artery has a whole lot of elastic in the tunica media, and that elastic will facilitate the movement of blood, because this is kind of an important concept to get here and understand, is that as that blood is coming out of the left ventricle with 120 millimeters mercury worth of pressure behind it, I mean, it's moving, and it's very, very forceful as it comes out of that ventricle into the aorta. And so if the walls of the aorta were just perfectly rigid, the blood would just hit it and stop. But because of this elastic fiber in there, the walls will give, like whoosh. So they kind of give, and then as they recoil, ooh, that it's going to move the blood on through. So it will enhance the movement and facilitate the movement of blood coming out of that left ventricle. Whoo, we stretch that elastic fiber, shh, we recoil, and the blood just moves right on through. Okay, so that's what conducting arteries are all about. We have to keep that blood flowing smoothly and propelled. Distributing arteries have more muscle in the smooth. More smooth muscle in the layer of the tunica media, and the smooth muscle will facilitate being vasodilated and vasoconstricted. As we move down to smaller yet arteries, we're down at the arterioles, and the arterioles are what precede the capillaries. So right before blood is released into capillaries, we have our arterioles. Okay. Once we reach the capillaries, that's where our exchange is going to take place. So here's a look at one of our conducting arteries, and you can see all the elastic fiber within the artery. I'm sorry, I just blanked there for a second. This is what we'd see under the microscope, which would help us differentiate structurally the difference between what an artery looks like and what a vein looks like. And you can see how the thickness of the wall of the artery helps it to maintain its shape, how thick the smooth muscle wall is, or layer is here. And the difference being, look at this vein, it looks, very close to being collapsed. Well, that's true. Because of the thinness of the layers in the walls of a vein, the lumen, if you put a vein next to an artery and from the outside they looked like they were the same size, actually, if you cut it open, you would see that the lumen of the vein is much larger than that of the artery. And that's because the walls are so much thinner. Well, when you have thin walls and a very large lumen, and the blood won't fill up the lumen just at rest. It's so most veins at rest are partially collapsed, and that serves as a nice sort of gotcha back situation, because if we start to exercise very quickly and there's a rush of blood returning to our heart, there's plenty of room for that blood within the venous system. So let's take a look at one of the roles of the arterioles. So here comes the blood from the arteries out, and keep in mind that this blood is going out to all the cells of our body, and that all of our cells are going to need the oxygen and nutrients and have their waste removed if they're living cells. And so as we get to the as we get to the capillary beds, the capillaries really are pretty much just like this mesh of vessels. They're so teeny, teeny, tiny. The walls of capillaries are not the same as the walls of arteries or veins. These walls are made up of cells, and the thickness is 1 cell thick. So the walls of these vessels are just 1 cell thick. And so when we get to the tissue, when the blood arrives at the tissue, it's going to like move through this mesh of capillaries. And this capillary mesh is so widespread within the tissue that every 2 cells or so there will be a capillary that passes through there, because keep in mind what has to happen here is that this is where the gases from the blood will exchange with the cells, and oxygen will be dropped off, carbon dioxide will be picked up, nutrients will be left at the cellular level, and the cells have to be able to readily get those things. So the capillaries are so widespread, again, very, very close in proximity to most cells. There's only going to be a couple cells in between each capillary. Again, capillaries are very microscopic. We're only going to see them under a microscope. So let's just take a look at how does our body decide, or how does it mechanically determine which capillary beds are going to receive the greatest amount of blood. Well, one way that we do this, we know that blood is going to be heading towards the most active cells. They're the ones that need what blood has to offer. All cells are going to be receiving blood, but we're going to send a little more in the direction of those that are in need because they're active. So actually what you would see off of all of these little trunks on the arterial are additional capillary beds that have an arterial aspect to it and a venous aspect to it, okay, throughout the arterial. So all over the arterials here, we're going to have additional capillary beds like this. Well, if we're wanting the blood to head out to, say, this capillary bed, because the cells here are much more active, as the blood comes through this arterial, the arterial has control over these what we call pre-capillary sphincters that can be closed. And again, they're not closed completely. All the cells are going to receive oxygen and nutrients, but they can be closed off in order to enhance the flow of blood to tissue where the cells are very active at this time. So here we have our blood that reaches these cells. Oxygen's dropped off, nutrients are dropped off, we pick up waste, and now we have used blood. Well, we know that used blood from the tissue now needs to head back towards the heart. So it just transitions into the venous aspect of the capillary bed. Okay, so these are venous capillaries. So they're going to be carrying that used blood. We're going to be heading back towards the heart. And the structure that the blood will enter after leaving the venous capillaries are the venules, which you can see, kind of comparable to the arterioles in size. And these venules will be fed by numerous capillary beds, and the blood will then go into larger and larger veins until it reaches the vena cava and then enter the heart. Let's take a look at some characteristics of capillary walls. We say there are 3 different types of capillaries. One being what we call continuous capillaries, and the cells in the wall of this, again, 1 cell thick. The wall is 1 cell thick, but the junctions between those cells are what we call tight junctions. And a good example of where we find this is in the brain, where we talk about that blood-brain barrier. Remember when the nervous system, we talked about that? Because we protect neurons, and we don't want neurons exposed to what might be in the blood. And so we keep the blood then in these capillaries. So that's a good example of continuous capillaries. Fenestrated capillaries, Well, between the cells that make up the wall, you'll find just openings. We'll refer to them as filtration slits, or just loose connections between those cells that make up the wall. Again, one cell thick. Sinusoidal capillaries. I refer to these as Swiss cheesy sort of capillaries because these capillaries have like gaping openings all through them, and where we find them is in a couple of organs where it's really important that the cells of that organ are able to access the contents of blood, everything in there, and so that access is given to them through these capillaries that have gaping openings in them, and again, very Swiss cheesy in nature. This is an example of a capillary that I would say this is more fenestrated, has some openings, and when we talk about fenestrated capillaries, you can see that really what's going to fit through the openings of the capillaries is just anything that fits. We're going to have the fluid from within, which we refer to that as plasma, and we know that plasma is going to have a lot of solutes in it, and so the plasma and its solutes are going to be able to pass through the openings in the capillary once it reaches the cellular level. Well, the things that cannot fit through these holes are things like red blood cells, white blood cells, platelets, which we'll talk about those, and things like large proteins. And that large proteins will then remain with the blood cells themselves, that we refer to as formed elements, will remain inside the capillaries. And any other larger molecules that can't fit through these holes. So again, anything that fits can come out through those holes. There will be a little bit of blood pressure pushing blood as it comes through the capillaries, and we call that blood hydrostatic pressure. And that little bit of pressure, we know it can't be much because it would just blow these cells right apart. So that little bit of pressure helps put a little bit of push behind this fluid, the plasma, to help facilitate its movement through the openings in the capillary wall. This is a diagram of the capillaries. And we're going to talk a little bit about what happens at the capillary level and how is it that the fluids move out and nutrients and gases move out or back into the capillary. What's happening there? So here's our arteriole bringing the nutrient-rich, oxygen-rich blood into the arterial end of the capillary. This is the venous end of the capillary, so blood will be entering that area and leaving the capillary through the venule and back out into the venous system towards the heart. So what we're going to look very closely at now are the pressures involved with moving substances out of the capillaries at the arterial end. When we talk about the movement of substances out of the capillaries, we talk about net filtration pressure. Now, I mentioned just in passing sort of hydrostatic pressure, blood hydrostatic pressure, and I said that's coming from the blood pressure, and we know that starts out at that left ventricle with the big gigunda push that comes out of there at 120 millimeters of mercury. Well, by the time we get all the way down to the capillaries, we don't have that much pressure left. In fact, we're looking at about 30 millimeters pressure behind the blood at this point, or even less, sometimes even somewhat less. But let's just work with these numbers. The blood hydrostatic pressure, we would say that it's pushing out, it's behind the blood, and it's pushing the fluid and anything that would fit through the holes, it's pushing out. And we would add this to interstitial hydrostatic pressure, which is just sort of a flow of the interstitial fluid that's actually kind of created by the movement of the fluid and substances coming out of the capillaries. So we've got this outward movement of about 33 millimeters of mercury, or pressure, yes, pressure measured in millimeters of mercury, sorry. As far as other pressures that we need to consider, we talk about colloid osmotic pressures. Well, in order to understand this, I think you need to have a little bit of a flashback to 2085 when we talked about osmosis. And we said that water will move across a semi-permeable membrane to the area where there's a higher concentration of solutes, because it wants to dilute those solutes and create somewhat of an equilibrium across that membrane. So if we look at substances that will create, I don't want to say a draw, a pull, but create circumstances where water will want to move in that direction. We look at the blood, well, like what's in the blood, and as I said, we have all these formed elements that are going to stay within the capillary. We have the red blood cells, white blood cells, platelets. We have large proteins. Those are we would look at that and we'd go, whoa, okay, that's the concentration of substances that we're looking at that will have, that water will be drawn to. The water will want to stay in the capillary because of solutes that are still there, because of the proteins, the larger proteins that are still there. The fluids in the plasma will want to stay here, and so that's going to kind of counter that pressure that's pushing out. And we say that there's a pressure of about 20 in as a result of that. So all we have to do is some simple math then. Oh, so we've got 33 out and 20 in. That means that the total is going to be 13 out, and that's our net filtration pressure, 13 millimeters mercury out. So since we have that pressure out, then on the arterial end of a capillary, we're going to get the movement of substances out into the cells. Okay, actually out into the interstitial fluid. Okay, so as the blood then is, well, what I wanted to point out, which I almost forgot, which is kind of mind-boggling, but over the course of 24 hours, and we think about our entire system, our entire body inside, the amount of plasma fluids that are going to pass out of capillaries at the arterial end is going to equal about 20 liters. 20 liters. That's 10 2-liter bottles. Okay, that's kind of mind-boggling. All right, so as what's left in the capillary moves into the venous end of the capillary, our pressures are going to change a little bit. And the key pressure that's going to change is our blood hydrostatic pressure. Because once the blood has been in the capillaries, the pressure is decreased even further. So we have far less pressure behind the blood, pushing the blood, once we're in the capillaries for a while. And so the blood hydrostatic pressure drops to about 10 from 30, and that's really the key difference. So when we look at what's happening now, we say, oh, well, we've got 13 out, and we've got 20 in, because that didn't change. Well, that gives us a net reabsorption pressure, a net of 7 in. All right, so what we're going to get at the venous end of the capillaries, we're going to get a movement of this fluid back into the capillaries. However, we're only going to get 17 out of the 20 liters are going to come back into the capillaries at the venous end. Now I want you to give some thought to what happens to that additional 3 liters, because 3 liters is a lot of fluid. And I can tell you we're not just going to leave it out in the tissue, because we know that leads to edema, and edema means swelling. So what happens to that 3 liters? I don't want to tell you because I want this to be kind of a cliffhanger. Um, and I want you to think about this a little bit. And of course, we're going to talk about it in class, so you better come to class and we'll fill in that particular blank for you. But kind of interesting stuff. So we're going to have this excess 3 liters of fluid, and this is systemically. This is not in just one little area, and it's over the course of 24 hours. The remainder, those 17 liters that flows back in, and the remainder of what's in the capillaries will then return to the heart through the venous system. Speaking of venous system, let's talk a little bit about the structure of veins. We talked about them being thin-walled and having large lumens, but we know that we just talked about when we're at the venous end of the capillary bed, that the pressure is very, very, very low. And, we had that powerful force, that push that came from the left ventricle to get the blood to the capillaries, but now it's kind of fizzled. There's not much there. And so we need to talk about how it is that the blood is going to move back towards the heart and to reach the heart through the venous system. Well, one of the mechanisms that's going to make that happen is that valves have, I always say that wrong. Veins have valves. Veins have valves. And so once the blood moves past a valve, the valve will close and it can't go back down. Okay, it can't go back down further and return to where it came from. So one of the things that's helpful in understanding how these veins work is that, they're positioned in between muscles and in between muscle and bone, and so they're typically quite deep. And so in order for these large amounts of blood to be returning to the heart, if you use a muscle, it's going to actually put pressure on the veins and cause the blood to move through the vein, and it's only going to move in one direction because the valve is closed, and so the blood will move and then the vein will close, and so the blood will move as in response to contracted skeletal muscles. Now, in addition to contracting skeletal muscles, because you're being active or you're running or you're going up and down steps, whatever, in addition to blood moving in response to that, there are always the respiratory muscles that are working as well, and they're also going to facilitate venous blood movement. Again, it's this muscle contraction working in conjunction with these structures that have valves, and so the blood will only move in one direction, towards the heart. When blood is when we're at rest, and we look at the distribution of blood, you can see that a much higher percentage of the blood is actually in the venous system. Again, veins, the lumen of veins are much larger. And we refer to this as a reservoir, and I think that's real kind of suiting because if we were to all of a sudden we're at rest and all of a sudden we initiate exercise, we initiate an activity, we know that there's going to be a surge or an increase in venous return of blood, and so it's good that we have additional blood within the venous system that can make its way to the heart rapidly and continue out to those tissues that are in need. So that is our reservoir. Again, it's the veins, venules that at rest will contain more blood than the other tissues. I always enjoy these two diagrams because you they're just a picture of what you know about the autonomic nervous system, the sympathetic and parasympathetic divisions. So you could say to me, you could describe to me in words what these pictures depict. And so let's just talk our way through this, because at rest you know what you would expect as far as the amount of blood that's heading towards different tissues in the body, because at rest would be considered what aspect of the what aspect of the autonomic nervous system? The parasympathetic division, right? Okay, so during exercise then, take a look at this. We're up to 17.5 liters per minute cardiac output, so we are definitely doing some work, and so we would be under sympathetic influence. We'll look at the difference as to where the blood is going to go. Obviously, it's always going to the more active tissues, especially in a sympathetic response. And so if we look at rest, parasympathetic, we know that digestive and renal are going to be quite active. We know that they're going to receive a fair portion of the blood that's in the arterial system, and that muscular will get sort of a moderate amount. Now, one of the things I want to point out is that the amount of blood that's going to the brain at any given time, whether you're exercising or whether you're at rest, that's going to stay pretty stationary, pretty much the same. Because, we protect that brain no matter what, and we're not going to allow that to fluctuate with any significance. Okay, so parasympathetic, at rest, cardiac output. Where's the blood going? Well, here we go. We know it's going to digestive and renal, to the muscles that might be mildly active, but so then we move into the sympathetic arena. Look where the blood goes. 12,500 mils per minute versus 1,000 over here in parasympathetic. Big chunk, big change. So again, blood is going to the active cells, the cells that are most in need. And look where we cut back. We know this. During sympathetic, we cut back from renal and digestive. We repress the activity there. We repress the amount of blood that's being flowed to those, that's flowing into those, the tissues of those systems. So I love this. So keep in mind when you look at this, that this is sympathetic, this is parasympathetic, and this is the distribution of blood that's occurring. This is another way of looking at that. If you're at rest, like Mr. Joe Belly Bob is here, you can see that the vessels to his intestines are vasodilated. All right, vasodilated, and so at rest, more blood heading into the digestive system and renal system, and not so much blood heading into the legs. Belly Bob's got his feet up and all of that, so reduced blood flow. But if you're being sympathetic mode out here playing soccer, there's going to be some vasoconstriction in the area of the intestines and the renal system. So we will have reduced blood flow to those areas, and as we know, dilated to the legs because there's a greater demand there. Okay, we're going to end for now, and we will continue with a different video.